Thank you, and I want to thank the organizers for the kind invitation to join you. I'm going to be talking about complement regulation and SARS-CoV-2, which is unfortunately the reason why we're not all in person. But this headline from the Washington Post kind of grabbed my attention. This was a little over a year ago, probably 16, 18 months ago. And why does it spare some and kill others? And kind of drew me in a little bit. And then I started to get a little bit more interested. Ed, I see you published one of these. And there was a plethora of articles coming out about SARS-CoV-2 and MERS and lots of coronaviruses tickling complement.
Rob, and sorry to interrupt, but could you share your screen?
You can't see this?
Your beautiful slides, not.
What about now?
Bottom.
Can you see this slide? I guess. Okay. So anyhow, that kind of drew me into this field, and there were also animal models showing that complement is deposited on a lot of the end organ damage. And also in humans who were succumbing to SARS-CoV-2, there was a lot of complement on the cell surface. So, you know, the question then became, where is this occurring? So you have this morass known as complement. And so what is this virus doing that is either activating or dysregulating complement and causing end organ damage? So the first thing we decided to do is I needed to learn about this virus a little bit. And I remember hearing everything about the ACE2 receptor, and everyone was focusing on the ACE2 receptor for the SARS-CoV-2 virus. And I started to look into it a little bit more, and yeah, that's how it gets in the cell. It's not really the receptor. What this virus does is bind to glycosaminoglycans on the cell surface. That's the original attachment, and that'll become very important as we discuss mechanisms later. It's focusing the virus on the surface of the cell that allows it to gain entry to the ACE2 receptor, and that is the entry receptor for the virus, but that's not the initial binding site. And that'll be, so remember that because it'll become more and more important. As more background, I want to talk to you about other disorders that dysregulate complement, and one of them is atypical hemolytic uremic syndrome, now known as complement-mediated hemolytic uremic syndrome. And what happens in this condition is patients are often born, they develop, sometimes they're familiar, but often sporadic mutations that occur in complement regulatory genes, and they're almost always in the alternative pathway of complement, which is the most important of the three pathways. And what happens is there's usually a trigger that upregulates complement, and then you have this uncontrolled complement activation on the cell surface, typically the endothelium, that leads to endothelial damage, fibrin deposition, and clotting. So these are the mutations that we frequently see in about 60% of patients with atypical HUS. They can be loss of function mutations in things like complement factor H, I, MCP, or activating mutations in things like complement factor B or C3. And these actually aren't really activating, they just prolong the half-life. But patients are born with this, they don't have but they aren't born with the disease, and it isn't until they encounter a trigger. And this accounts for the variable penetrance, and it's usually some inflammatory state, big surgery, third trimester pregnancy, an autoimmune condition, etc., that sets off this overwhelming complement activation on the cell surface and triggers the disease process. When attending on our hematology service, we often encountered many patients with thrombotic microangiopathies, but we couldn't tell whether they were complement driven or not. So we wanted to really develop an assay that would get at this. And I'm going to spend a little time on this assay because most of our complement assays really aren't very helpful clinically. They're based on red cell lysis assays of, using rabbit or sheep erythrocytes, which aren't really, they're a little too sensitive because a single membrane attack complex will lyse a red cell. Or we're measuring complement levels in the serum, and they can be down because of lack of production or consumption, just like many coagulation factors. So we wanted to design an assay that tested how a nucleated cell regulates complement, and this was called the modified Ham test. It's named as such because I flipped around the typical Ham test. Some of the older people will remember in the room that was used to diagnose PNH. That was described in 1937. But briefly, the way this assay works is we take a nucleated cell line and we've knocked out pig A, basically to knock out terminal complement. And then if we add control serum, we can, without the terminal complement regulators, we can measure how the cell regulates complement on its cell surface. And if it has good complement regulation, the cells will remain alive and they won't have complement-mediated death as measured by a WST1 assay. But if they have problems that are in the serum, that such as, with factor H or factor I or C3 or factor B, and they can't regulate complement, they will lead to membrane attack complex formation. And if enough of it occurs, the cell will die and it won't release the WST1 dye. We do this with a control of heat inactivated serum to prove it is complement dependent. And this is our modified Ham test that we use, and I'm going to be discussing going forward. And if we see 20% or more killing, at least in thrombotic microangiopathies, that was our threshold for a positive test. We can use a variety of things as a positive control. Shiga toxin is one of the things we often use, but we can also use neuraminidase or sialidase, introduced by Dr. Brown, which cleaves off the sialic acid residues, which are important for factor H to bind to. And this is just to show you that when we took serum from either atypical HUS patients or TTP patients, where have a totally different mechanism for getting a thrombotic microangiopathy, this is a deficiency of Adams TS13. We can really distinguish the atypical or complement-mediated hemolytic uremic syndromes from TTP or normal serum, unknown TMAs, or sepsis or anything like that. So I also want to show you an assay that we developed to go along with this. It's a flow cytometry based assay because we wanted to show, the modified Ham test requires the cell to die, but we can look a little earlier in the process and measure C5b9 expression on the cell, not in the serum. The serum, it's very soluble C5b9 can be very unreliable. This is on the cell surface. So what we did here is what we call a C3 loading assay. And what we do is we basically add the components of the alternative pathway, C3, factor B, and factor D. And then after we add those factors, all you can do is get to C3b, but you can't get to terminal complement. So we add human serum with and without inhibitors, and then measure the cell kill. Once we add the serum, all the other factors are there. So here you can see our modified Ham test. We're measuring non-viable cells. This is the threshold of what we call positive. And this is just one cycle of adding C3b to the cell surface, 2 cycles, 3 cycles, 4 cycles. You can see the more C3b we add to the cell, when we add the serum, the more cell killing till we can get to the point where we can kill every cell on the plate. And we measure this in a plate reader. We can also show that when we do this in EDTA, which chelates calcium and magnesium, we don't see kill. Here's this is with a factor D inhibitor that is specific for the alternative pathway. We can block it, and if we use a C5 inhibitor, which is terminal complement, we can block the cell killing. So that just is another assay. And then we can look at this and show this by flow cytometry. So there's a lot of panels here. So focus on the top. So this is normal human serum that is heat inactivated. So all the complement is killed when in the serum. So we here is, cycle 1, cycle 2, more C3, and cycle 3, almost all the cells are loaded with C3. On this axis, we're looking at C5b9. We're not seeing any C5b9 staining because we're adding heat inactivated serum. There's no complement there. Here's normal human serum in this second row going across. And as you can see, you get to a certain threshold of C3b and you start flipping over. But when you get a lot of C3b on the cell surface and you add the serum, almost everything flips over to C5b9. And that's exactly the same concentration that we're put in when we saw the cell killing in the MHAM. Now, here is when we're adding a factor D inhibitor in the third row going across, the second from the bottom. And again, you see the C3 going up, C3b going up, but with the factor D inhibitor, because you're blocking the alternative pathway, you're not able to get to terminal complement, and you're not seeing C5b9 form. And same thing with C5 depleted serum, or if we add a C5 inhibitor. So now we have a flow cytometric assay to go along with our modified HAM test that correlates very well. All right, so let's talk about SARS-CoV-2, and I got involved in this, about a year and a half ago, most of the labs were closed down to research other than COVID research. So we thought, let's take a stab at this. So I was quite interested in the spike protein because that was the major protein that was on the surface of the cell. So I couldn't get serum. It was impossible to get serum from these patients early on in this pandemic for all sorts of IRB reasons and logistics. And so I decided, let's buy recombinant components of the virus. So we bought the S1 and the S2 component of the spike protein. We bought the N protein, which some people were saying is in China that was activating the lectin pathway. And we also bought the spike protein from another beta coronavirus that is very related to SARS-CoV. This virus, OC43, accounts for 15% of the common cold. Doesn't cause severe end organ damage. There's almost no more mortality from it. Causes basically the common cold, but nothing else. So what we did is we just took normal human serum, and you can see here's our modified HAM test. And normal human serum, typically we see, less than 10 or less % cell kill. Normal human serum that's heat inactivated, no cell kill at all. And then we just did a dose response curve with the S1 component and the S2 component. And you can see there was a nice dose response as we got up to, especially when we got into the 10, 20, 30 microgram range, we started seeing lots of cell death in our modified HAM test. The N protein did nothing. And the spike protein from OC43 did nothing. So this interested, this drew me in. And then we wanted to make sure, heat inactivation, but we wanted to make sure that this was complement dependent. So we added the spike protein in the presence of a C5 inhibitor and S1 and S2, you can see, completely blocked the killing. So this was telling us that somehow these spike proteins are causing complement activation, dysregulation, whatever, and killing cells in a complement-dependent manner that was preventable by complement inhibitors. But how? So let's take a little bit of a deeper dive into the alternative pathway. And just as a refresher, the alternative pathway is always on. It's not really activated. It's our cells are constantly being challenged with C3b, and that's because in the fluid phase, C3 can be hydrolyzed just with water. There's a thioesterol bond that is cleaved, and that allows factor B to bind to C3, and factor D can cleave it to generate the fluid phase C3 convertase, which can also cleave C3, generates C3b, and again, this sets off the whole amplification loop in the alternative pathway and continually bombards the cell with C3b molecules on the cell surface unless there are important inhibitors. So if you have an activator surface, such as a bacteria, PNH red cell, for example, these surfaces, there's nothing to regulate or very little to regulate this form of the alternative pathway. And it's going to go all the way to the membrane attack complex, because when you get two C3b molecules to one big B little b, that's the C5 convertase that cleaves C5 into C5a and b, and that allogrammizes with C6, 7, 8, and 9 to form the membrane attack complex, and it forms a pore in the cell surface. Inactivated or inhibitory surfaces have complement regulator proteins on them. And these are things like complement factor H, complement factor I, MCP, all the genes that were mutated in atypical hemolytic uremic syndrome. So they don't really cause activation of the alternative pathway. What they really do is they lead to dysregulation of the alternative pathway. So under normal circumstances, you should be able to control complement on the cell surface. If you have mutations in genes like these, factor H, factor I, etc., you can turn an inhibitory surface into an activator surface. So that's what we wanted to probe and to say, hey, where is this happening? Which pathway? Is it alternative? Is it classical? Is it lectin? And if it's alternative, is it fluid phase? Is it cellular phase, etc.? So in this set of experiments, we used our flow cytometry assay, and here we're looking at C5b9, or the membrane attack complex, not in the serum, on the cell. And here's EDTA. Again, there's nothing with normal human serum because you need calcium and magnesium for complement, just like you do in coagulation. The alternative pathway only needs magnesium. This is normal human serum. You see some small amount of deposition, but when we add the spike protein, again, we use the 20 microgram amount. We see, over 50% of the cells have C5b9 on the cell surface. If we add a factor D inhibitor, which is specific to the alternative pathway, or a C5 inhibitor, which blocks all pathways at C5, you can block the C5b9 deposition. Now, in this flow assay, I'm looking at C3c deposition. So these are C3 fragments on the cell surface. And again, they go up when we add the S1. We are able to block them with factor D inhibitor, but no effect when we add a C5 inhibitor. And we see the same thing with the S2, even more pronounced. We see the factor D inhibitor blocks the C5b9 deposition, but anti-C5 or an eculizumab-like compound doesn't do anything. The C3 is still on the cell. And this was the first hint here that this is alternative pathway. We then wanted to determine, is this happening in the fluid phase or is this happening on the cell surface? So that was the next set of experiments. And so what we did is we measured big B, little b. So why did we do that? Well, when C3b lands on the cell surface, factor B binds to it, and then factor D cleaves it, and you get C3b, big B, little b, but that has a half-life of about 90 seconds, and it comes off into the supernate. And you can measure that. So we did our BB assay in the presence or absence of cells. So we just took normal human serum, okay, and with cells and without cells and measured BB. But when we added the spike protein, only did we see an increase with the cells present. So we saw it with the S1 component and the S2 component, but with no cells, we saw no increase, actually a slight decrease in BB. And that told us that this process of dysregulation of the alternative pathway was happening on the cell and not in the serum. So then what we did is we wanted to see, hey, can we block this? So over here, you can see again the normal human serum. That's the baseline amount of BB. We add the spike protein, it more than doubles. We add a factor D inhibitor, no BB forms. So we're blocking the dysregulation. We're blocking the alternative pathway of complement. When we added a C5 inhibitor, no effect on BB. So this was really telling us that this is alternative pathway. And same thing with S2. It was completely blocked by the factor D inhibitor, but not by the C5 inhibitor. So that was telling us that C3b is landing on the cell and somehow being inactivated or leading to dysregulation, where you're leading to this amplification loop. You can block it with a factor D inhibitor, but downstream you can't block it because the dysregulation has to be upstream. Okay, so again, thanks to Dr. Brown for introducing sialic acid, because I was sure that it was going to be sialic acid. Reason being is factor H binds to sialic acid, and for factor H to function, which is a critical negative regulator of the alternative pathway, it needs sialic acid on the cell surface. We did a bunch of experiments with knockouts, etc. Suffice it to say, it wasn't sialic acid. But factor H binds to a cell in multiple ways, and it competes with factor H for binding to factor B. It accelerates decay of the C3 convertase, and it's an important cofactor for factor I. You can think of factor H as like a string of pearls, and it binds to C3b, it binds to sialic acid, but it also binds to this C6, 7, 8 region. And there are human diseases that are associated with that. Atypical HUS, the mutations tend to be in the terminal region or sometimes this proximal region. But there's another disease called age-related macular degeneration, where the defect is in C6, 7, and 8. And for factor H to function perfectly normally, it needs to make contact with all three of those points. So then we did a competitive binding experiment. And we took saturating doses of complement factor H and heparin beads. Why heparin beads? Because I told you that spike protein binds to heparin sulfate, glycosaminoglycans. And we wanted to see whether the spike protein was binding to the heparin sulfate and outcompeting factor H so that it couldn't bind to the cell. And sure enough, that's what we found. And so what we did is we incubated the spike protein with complement factor H overnight. We centrifuged, washed, eluted, and then did a Western blot. And what you can see is that when we added the spike protein S1 or S2, we saw a marked decrease in binding to heparin. But when we did this with OC43, we saw no such blocking. So this was telling us that the spike proteins are competing with factor H for the same binding site, heparin sulfate, on the cell surface. And so we have this model of what's going on, and you can imagine if you get SARS-CoV-2, you're going to tip the balance to loss of function in factor H. And then you can, if you have further amplifying hits, you're going to get more end organ damage and endothelial damage, and often sometimes even TMAs that you see with SARS-CoV-2. So what really struck us was a paper that came out around the same time where they were looking at risk factors. This was done out of Columbia for developing severe COVID. So, and what they found with over 11,000 patients was that a history of macular degeneration was the biggest risk factor for mortality and intubation, that was even higher than effects explained by age, sex, or history of smoking. The interesting thing is that this polymorphism was described in 2005, and this is the exact same region where complement factor H binds heparin and heparin sulfate and C-reactive protein. So this spike protein is binding in the same spot where individuals with age-related macular degeneration tend to have base changing polymorphisms. So then we wanted to see what happens. Now we're into this, and we finally get serum from SARS-CoV-2 patients. And again, this is our modified Ham test. Here's normal human serum. Here's sialidase. Here's shigatoxin. These are our positive controls. And then we had 59 patients that were either, that everyone was hospitalized. These patients required supplemental oxygen, high flow oxygen. These patients were vented, and there were 2 that died. And again, we saw a correlation with severe disease in the modified Ham test in the serum. When we looked at BB levels, every patient who had SARS-CoV-2 had markedly elevated BB levels. We saw some more of it in the severe cases, but all of them had elevated BB, suggesting that the alternative pathway is involved probably pretty early. We then got interested and we said, hey, these mRNA vaccines, are these going to be safe? Because if they're increasing the spike protein, is it enough that's going to be able to activate complement and cause problems? Well, in normal controls, we found that wasn't the case. We took 5 healthy controls and we measured them at baseline immediately before their vaccine, and then within 24 hours after dose 1 and within 24 hours after dose 2. None of them had a positive Ham test. We did see a slight increase in C5b9 deposition, but not that much. But when we looked at BB, there were 2 healthy controls where they doubled their BB, and these were the 2 that had the most severe side effects from their second shot. So it basically says it was reassuring that these mRNA vaccines are going to be safe, but there could be a population, especially patients with underlying complement dysregulation, who may have trouble. And indeed, that was the case, and this was published in Blood just recently. These are PNH patients that got vaccinated, and I know it's hard to see here, but the first patient was not on any complement inhibitors because he had very mild symptoms from his PNH. But after his second vaccine, developed microvascular thrombosis within days and ended up being hospitalized, needed anticoagulation, and then had to start on a complement inhibitor. These are patients that are already on complement inhibitors. This one dropped his hemoglobin 3 grams. This woman also dropped 3 grams, had to get 2 units in the emergency room. This one dropped 4 grams, and bilirubin rose to 7.1, but didn't get hospitalized, and this patient did fine. So we find about 40 to 50% of our PNH patients have severe breakthrough hemolysis when they get these vaccines. Still better that they get vaccinated, but they should be aware of it. So this is kind of the model that we're developing here, is that, and we think that early on it's the spike proteins that change a inactivator surface into an activator surface and can cause endothelial damage. And then when you have additional hits, such as 3rd trimester pregnancy, complement-associated mutations, thrombosis, infection, obesity, diabetes, in things that cause inflammation in a high CRPs, you can amplify this condition. And so I'm going to stop there and say, that the spike proteins bind to heparin sulfate, not primarily to the ACE receptor. They eventually get there. And that blocks the function of factor H. Factor D inhibition and C5 inhibition block C5b9 deposition and death in the modified Ham test. But factor D is a much better blocker because it blocks upstream. C5 inhibitors don't block upstream. The N protein and spike proteins from other coronaviruses don't bind cells well and don't turn this inactivator surface into an activator. And COVID-19 is a complementopathy and may be amenable to anti-complement therapy. Proximal complement inhibitors are likely going to be better, and there is currently a randomized controlled trial by NIAID comparing a factor D inhibitor with remdesivir to remdesivir alone. Those results are pending. And but if you're going to do this, you're going to have to start early in the disease process before you have too much endothelial damage. And then watch out for severe vaccination reactions in patients with underlying complement disorders. And I will stop there and just thank a lot of the people that did this work listed here and shown here. So thank you very much.
Thank you very much. We have time for some questions.
Definitely. Hi, Robert. Ed Conway here. That was fantastic. It's a lesson on how to teach complement. Can you hear me? Okay, you're muted now. I do have a question about the antibodies, the passive immunization, the Eli Lilly or whoever gives these antibodies. Have you ever looked at those? Can't hear you.
Trouble.
We lost audio of Robert.
I'm going to throw on a set of
headphones.
headphones.
Oh, there you go.
Okay.
You're back.
Can you hear me?
Yep.
Okay.
So the antibodies against that have been used in treating patients, do they have any effect in your Ham test in the presence of the S protein, the spike proteins?
So specifically like eculizumab and danicopan?
No. I meant the anti-SARS coronaviruses from patients that have been used to treat. The ones that Trump used.
Oh, no. So no, we haven't tested those. I don't have access to those antibodies.
Questions?
I have a question.
Oh, Dan.
Yeah, thank you very much. That talk was really interesting. I'm kind of curious about the PNH patient response to the vaccines, and I'm wondering whether this isn't just the formulations of the liposomes that are used to encapsulate the mRNA, and whether they're not just complement activating, which is normally controlled by somebody with adequate amounts of complement regulatory proteins, but when you get in these PNH patients, it just doesn't work.
Yeah, so it's a fantastic question, and I didn't get a chance to go into the detail that we went into in the paper. We looked on PNH red cells, and it's not because the spike protein is binding. The spike protein doesn't bind to red cells very well because red cells don't have much heparin sulfate on it. It is, as you suggest, cause the inflammatory response from these vaccines that PNH patients are susceptible to. We can see PNH patients break through to influenza vaccine as well, but not like this. They really break through, and it's just due to a general inflammatory state. On the other hand, we have seen, and there are several reports of patients with atypical HUS that were in remission, come out of remission with these proteins. And with these mRNA vaccines.
I have a second question, which is, I'm just curious, have you ever taken just a culture of live virus and incubated them with serum to see if the virus itself is complement activating?
We haven't. I mean, just the restrictions around this is all with the proteins, and we haven't done it with a live virus.
Hello. Robert, can you make any comments about why eculizumab presumably failed? I mean, I guess there's multiple reasons, but maybe you can make some comments.
I mean, first of all, the studies are very small. Secondly, they were giving it to patients that were on high flow oxygen or ventilated. And for complement to work in this condition, I think you're going to have to give it pretty darn early. Because think about it, you get infected with the virus, and what happens is you build up the spike protein, and it takes about 7 or 10 days before you get to the point where you're pretty bad, where you get hospitalized. And then it might be 12 to 15 days before you're on high flow oxygen. You may have cleared the virus practically by then. Now you're just, you have secondary infections, you have comorbidities, you have all these other. So I think if you wait till someone is on high flow oxygen or has an ARDS-like picture and give them complement inhibitors or any therapy for that matter at that point, with the possible exception of steroids, you're not going to have a huge impact. And that's going to be the trouble with these inhibitors. You're going to have to pick the group that is. You don't want to give it so early because these are such expensive drugs, and you don't want to waste it because most of these patients are going to do okay. But you want to be able to get the patients that are going to be severe and get them blocked early. That's the only way a complement inhibitor is going to work. You do it too late, it won't work. The other problem with C5 is that there is a bypass to C5. When you coat cells with lots of C3b, you can get C5 to take on a C5b conformation without the C5 convertase. And this is recent data from within the past year and a half. So that's why people have that's why PNH patients have pharmacodynamic breakthrough when they develop inflammatory states or infections. They have intravascular hemolysis in spite of blocking C5. So you really want to be upstream.
Last question. Have you looked at patients, chronic COVID, the long haul syndrome in terms of ham test in these patients?
It's, I don't know what it's like up there, but it is really hard to get patient samples from these patients. And I wouldn't expect the long haul patients to be complement mediated. I think complement is a very early trigger. And I think there are patients that have some genetic susceptibility where you're in the process of doing a next gen panel to look for complement associated mutations. Because I would predict that just like age-related macular degeneration, if you see someone with a factor H variant that's functional or a C3 and they get COVID, look out.
So maybe while you guys are sorting that out over there, I'll just ask one more. I'm curious about what you think about the intersection of the role of complement in COVID and the ABO blood groups. Do you think there's an intersection there?
So I don't, yeah, I'll just leave it at that. No, I don't think there's an intersection.
Okay, thanks. And there's one more question over here. Ed?
Yeah, this is Ed Prysdale. There's a question that came from someplace in the cloud from our colleague Chris Overall. He says, thanks very much for a great talk. And do you have any evidence that heparan sulfate is taken up with COV2 on cell entry? He knows that spike glycoprotein binds galactin-8 in the cell as a virus sensor targeting the virus for autophagy destruction. which the virus evades by 3-Cl-Pro cleavage of Gal-8. And they also know that thiodigalactoside blocks this. So do you have any comment on heparin sulfate uptake into the cell?
I don't. It's an interesting question. And as far as I know, heparin sulfate isn't taken into the cell, but I don't know the answer to that question. So yeah.
Thanks very much.